morning, everybody. Thank you for joining us today. We will get started in just a moment here. All right, hello everyone, and welcome to today's keynote webinar titled the NIH Microphysiological Systems Program, Tissue on Chips for Safety, Efficacy, and Precision Medicine Studies. This webinar is a part of the 7th annual event in the Cell Biology Virtual Event Series. I am Sydney McNeil of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you happen to have any trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Now let's get started. I now present today's speaker, Danilo Tagle, Director, Office of Special Initiatives, National Center for Advancing Translational Services, National Institutes of Health. For a complete biography on our speaker, please find it on the left of your screen. Danilo, you may now begin your presentation.
Thank you, Sydney. Thank you for the kind introductions, and certainly thank you to the organizers, to Melanie and Lynn, for the invitation to present to you this morning. So I'll be talking to you about tissue chips or microphysiological systems. And this is the outline that I hope to accomplish this morning, to tell you a little bit about NCATS, the National Center for Advancing Translational Sciences at the NIH, the translational problems we're solving, and what tissue chips are essentially used for in terms of toxicity and safety studies, disease modeling, precision medicine, and how we're building confidence in the use of this technology, and then summarize with a few future directions. So let's start by me telling you about the National Center for Advancing Translational Sciences, or NCATS, here at the NIH. This is a new entity within the NIH. It was established about 11 years ago in 2012 by the United States Congress, with a different mission than the rest of the NIH. We're primarily established or stood up by Congress in order to catalyze generation of innovative methods and technologies that seeks to address the valley of death, or things that would hold back translating basic discoveries into applied science. So in terms of translational problems, here's the biggest problem we're facing in terms of drug development. The percentage of drugs entering into clinical trials has a very low success rate of 10%. So it's a high attrition rate of 90%, of which 55% fail due to lack of efficacy, and 28% fail due to toxic effects. So if you look at the chart on the right-hand side, you'll see that for various indications, whether it be hematology and infectious disease, we tend to do well at around 26.1% or so. But for indications like oncology and psychiatry, we're in the single digits of 6.7 and 5.1%, with the average of about 9.6%. So that is a very poor success rate in translating candidate drugs to go into the clinics. Moreover, it takes an average of 10 to 15 years at a cost of $2.6 billion per drug to come into market. And the reason for that is primarily the current tools that we're using, like 2D cell culture systems and animal models, do not always predict the human response. And that is shown here on the bottom chart, where you see poor concordance of toxicities within human and various animal species. In addition to the low predictiveness of the animal models and the tools we're using, more and more of the therapeutic modalities being developed in the pipeline are tending to be more human-specific and personalized, where it be small molecules, human-specific antibodies, protein therapies, of course, antisense oligonucleotides, and somatic cell gene editing technologies, cell therapies. They all tend to be human-specific and will require human-based or human-relevant model systems. And so back in 2012, when NCATS was first stood up, we formed the NIH Tissue Chips Consortium. And this is sort of like the landscape of all the activities that we have done over the past 11 or 10 to 11 years or so in terms of getting microphysiological systems established and developed, starting with partnerships with DARPA, a few pharmaceutical companies, AstraZeneca, GSK, and Pfizer, and then launching a program, but that first started looking in terms of, can we develop tissue chips for safety studies. This was then followed in the next 5 years, from 2016 or so, looking at deployment of microphysiological systems to look at efficacy or disease modeling. And now this is just an overview, so I'll be going into detail about aspects of this landscape. And then the most recent iteration of tissue chips is in application to precision medicine. And then also in terms of how we build confidence. And just to let you know that the partners that we have all throughout this program would be the FDA from the beginning of the program, and then in 2014 or so, working with a number of pharmaceutical companies from the IQ Consortium, Microphysiological Systems affiliate. So let's dig deeper in terms of what tissue chips are. So the NCATS tissue chips essentially are in vitro 3D culture systems that takes into consideration advances in microfluidics, stem cell biology, and bioengineering, and uses human-derived cells and tissues, whether it be primary cells or induced pluripotent stem cells. And the goal is to mimic basic organ function, as shown here. So if we're trying to mimic the lung, going back to the basic functional unit of the lung, which is the alveoli or the air sac, and this is sort of like the rendering of what a chip would look like, a cross-section of a chip, where the blue would be the lung epithelial cells separated from the bottom vascular endothelial cells by a thin elastic PDFs membrane that allows it to be stretched. And then cyclic vacuuming then allows the cells to actually undergo stretch. So biomechanical cues are an important feature of these microphysiological systems. And they're on the top of the lung epithelial cells would be air. And then in the bottom of the vascular endothelial cells will be media flow. So in some ways, recreating the air and liquid interface. So that is an important feature of microphysiological systems, would be microfluidic flow that mimics the perfusion of the system, as well as biomechanical cues, and of course, human relevant cells and tissues. So the goal is to recreate a number of 10 major organ systems in the body, from circulatory all the way to urinary system. And so what that looks like right now, this is actually the lung on a chip that is now commercialized by a company called Emulate. And then there are also platforms that contain multiple organs on chips that are all interconnected by microfluidic channels, such as this 5-organ chip that is manufactured by Hesperos. To give you a little bit more detail, there will be a video that should be playing soon in terms of showing the liver on a chip. And this would in some way show that human liver cells, hepatocytes, and the non-parenchymal cells are being seeded into liver chip and undergoing perfusion in this system. The next question would then be, once these chips are actually developed, are tissue chips better predictors of human physiological response? So just to give you an illustration, I mentioned there were a number of organ systems that we're trying to mimic. I'll focus on the liver for now. And so, as you know, the liver is responsible for concentrating and metabolizing a number of drugs and medications. And because of that, drug-induced liver injury, or DILI, is the most common cause of acute liver failure. And adverse drug reaction because of liver injury is a major source of failure of candidate drugs, resulting in hospitalization, liver transplantation, and even death. So just to show you an illustration of what the liver chip looks like, this is a cross-section of what the liver chip would be, similar to the lung on a chip that I showed you. So there will be representations of the parenchymal channel, as well as the extracellular matrix shown here, and most importantly, a very mixed ratio of cells consisting of hepatocytes, stellate cells, Kupffer cells, and endothelial cells that mimic the makeup of a liver sinusoid. In terms of illustrating how this is used, so in looking at a reference compound, phenyluridine is an antiviral agent that was developed for hepatitis B. When it was tested in rats and dogs, which are the typical animal models used to pre-screen drugs for safety, it was approved because by the FDA because it did not show any safety signal from the rats and dogs. But unfortunately, in phase 1 clinical trial, 5 of the 15 patients treated with phenyluridine actually died, causing the FDA to pull out phenyluridine from testing. So the question then would be, would the human liver on a chip have been able to predict the toxicity that the rat and dog models did not? So in order to do that, a rat liver on a chip, rat cells were put on the liver on a chip, as well as human hepatocytes on the chip, and then treated with various doses of phenyluridine from 0, 1, 10, and 30 micromolars. And as can show here on the top micrographs, showing that when stained with Nile red to indicate steatosis or deposition of fatty molecules in the rat, there's no yellow staining altogether, but in the human liver on a chip shows an increasing dose-dependent increase in steatosis or fatty deposit, showing a dose-dependent toxicity in the human liver on a chip. And this is quantitated on the right-hand side, showing again that the rat liver chip, even at various increasing doses, did not show toxicity, whereas clearly in the human liver on a chip recapitulates the toxicities that were found in vivo in the human trials. Now, to further look at the functionality of this chip, one of the common indicators of liver function is albumin secretion. So if you look at the rat liver on a chip, even at increased dose, it continues to secrete albumin whereas the human liver on a chip, an increasing dose treatment of phenyluridine shows a dose-dependent decrease in the secretion of albumin. Well, likewise, one can also look at indicators of liver injury markers such as miR-122, glutathione S-transferase, and keratin. In this particular markers, again showing an increase in this liver injury markers that is also dose-dependent. Now, this is only one compound that was tested, but this was followed up by a number of by a study looking at more compounds. So in this case, looking at 22 hepatotoxic compounds, just like phenyluridine. These are compounds as shown here and listed in the bottom. These are compounds that have gone through screening with animal models, have shown to be safe with animal model data, approved by the FDA, and went into the clinics. But unfortunately, collectively, more than 200 patient deaths, resulting in more than 200 patient deaths and 10 liver transplants. So again, these 22 compounds were withdrawn. And then when the human liver chip was used to identify if these toxicities could have been predicted, the human liver chip showed an 87% sensitivity and 100% specificity in being able to predict the drug toxicity, which is certainly a lot better than the model system that was used that only showed a sensitivity of 47%. Next up, in terms of disease modeling, again, the goal here is to recapitulate in vivo function. And since we're doing disease modeling, we have to be able to show that the chip can also, the tissue chips can capture both normal and disease states, be able to show pathophysiology, and of course, over the course of treatment, has to have stable phenotype over weeks and months, and also be able to reflect multi-organ pathology and organ crosstalk. So in this particular area, NCATS was able, with some partners at the NIH, were able to fund a number of different projects looking at a number of diseases from Alzheimer's disease and Parkinson's disease, which are neurological disorders, to some that are cancer-related, such as radiation-induced xerostoma, that is radiation treatment causing dry salivary glands, looking at rare diseases of the heart, such as Barth syndrome, rare diseases causing vascular malformations, such as hereditary hemorrhagic telangiectasias. So you can see here from this list of different projects that NCATS have funded, looking at common and rare diseases, as well as complex and single Mendelian disorders. The most complex, of course, being able to look at type 2 diabetes, with a number of interconnected organ systems such as pancreatic islet cells, liver, adipose tissues, and the gut. To show you the effectiveness of tissue chips in being able to be used for disease modeling, and especially in this particular case, looking at rare disease, we're looking at a very rare disease called chronic inflammatory demyelinating polyneuropathy and multifocal motor neuropathy. This is an autoimmune disease, affects very few individuals, and so there are no animal models that actually exist at this point. But using human primary Schwann cells and induced iPS-derived induced pluripotent stem cell-derived motor neurons to seed them on a chip to form a neuromuscular junction. This chip was then treated with serum derived from patients. In this case, 15 patients, as shown here on the chart on the right-hand side. Each micrograph is a serum, is a chip treated with a serum from a different patient, so 15 patients altogether, and stained to show that there is complement activation because of this treatment of the sera, just so in some ways recapitulating the autoimmune response in these chips. And then these chips were then used as a way to screen for candidate drugs. So in this case, one candidate drug that was taken from a repurposed drug, so previously FDA-approved drugs, TNT005, which is a monoclonal antibody that inhibits C1 protease, was then used to pre-treat the neuromuscular junction on a chip, and then again subsequently treated with patient-derived serum. And then in this pre-treatment of TNT005, the complement activation was not possible or did not happen because of the pre-treatment with TNT005. Shows the effectiveness of using this as to model not only the disease, but also as a way to screen candidate therapeutics. So in this case, TNT005 is now undergoing phase 2 clinical trial that is sponsored by Sanofi. One of the important things too that we could do was to use tissue chips to develop model systems for in response to the opioid crisis. Prior to the pandemic, the biggest health crisis that our nation faces was certainly the opioid crisis. But there are also no effective model systems for looking at pain circuitry. And so we were able to deploy tissue chips with congressional allocations through the HEAL program, HEAL standing for Helping End Addiction Long Term, to develop both models for sensory circuitry, pain circuitry, as well as the reward dopaminergic pathways, and to develop model systems for pain and opioid use disorder. Of course, with the pandemic, we were able to also use additional funds to be able to develop model systems to look at COVID-19 infection, because as you know, only a hamster and ferret were really able to show to be able to be infected, but they also did not show the full pathology, but the tissue chips did. And just to illustrate that, this is an example of, again, repurposed drugs. In this case, 7 antiviral compounds: chloroquine, amodiaquine, arbidol, dorimifene, verapamil, all treated with HA7 cells, infecting HA7 cells with SARS-CoV-2 pseudoparticles, and showing, at least in a 2D cell culture system, that these 7 candidate drugs would have been effective as an antiviral agent. But the question is, do you advance all 7 into the clinics? And so in some ways, the 3D, three-dimensional nature of a human airway chip was able to show which ones are actually going to be more effective. So in this case, using, again, the SARS-CoV-2 pseudoparticle, but in this case, instead of 2D cell culture system infecting 3D human airway HA7 cells on the chip and showing only amodiaquine and theramifene as actually being more effective compared to the rest. So certainly saving a lot of time and resources and moving things quickly. So amodiaquine is actually also now in human trials as an antiviral agent. One of the exciting things that we're also doing is to model age-related disorders. As you know, to model aging takes years, if not decades, to induce in animal models. And so there are a number of changes that happens as a consequence of aging, including musculoskeletal deterioration leading to osteoarthritis, osteoporosis, RA, and sarcopenia, or muscle wasting. That also includes GI conditions such as decreased gut motility, cardiovascular changes like hardening of the arteries, respiratory changes, renal stone formation, and immunosenescence or decline of the immune system. And so many of these things are associated with aging. but as I mentioned, takes a long time to mimic on model systems. But as it turned out, in looking at the clinical literature and working with NASA, the National Aerospace Science Agency, showing that astronauts, when they fly into low Earth orbit and exposed to microgravity, there are certain physiological changes that happens that resembles aging, such as it stays from 3 to 6 weeks. They undergo bone resorption, muscle atrophy, or sarcopenia. They also undergo cardiovascular deconditioning, GI disturbances, and anything that happens long term also leads to declining immunity and renal stone formation. But the important thing is that when the astronauts return back to Earth within 2 to 6 months, they revert back to normal physiology. And so, excuse me, and so in some ways it gives us the opportunity to ask the question, what are some of the molecular instigators or changes that happens that allows physiological changes to happen within a short period of time, from weeks to less than 6 months, that normally would have taken years or decades to happen here on Earth, in terms of the aging, and then more importantly, when we when the astronauts return back to Earth, what are some of the changes, molecular changes, that allows them to revert back to Earth. And so it allows us to be able to identify molecular signatures that might be important to mitigate or slow down the effects of aging. And so what we did then was partner with NASA, the Center for Advancement of Science in Space, or CASIS, and the International Space Station National Laboratory, and be able to deploy tissue chips in space, essentially to model age-related diseases under microgravity, so that we can develop countermeasures and interventions to mitigate or slow down the effects of aging. So we have projects on looking at immunosenescence, changes in blood-brain barrier permeability, changes in the mobilization of immune cells as a consequence from the bone marrow to the lung as a consequence of infection, looking at osteoarthritis, kidney stone formation, cardiac dysfunction, muscle wasting, as well as gut inflammation with changes in the microbiome. An important thing that happens too, in a consequence of working with partners such as NASA and CASIS, was that the improvements in engineering and automation. So here on Earth, when we run experiments on let's say on kidney on chip, looking at renal stone formation, it requires a substantial amount of instrumentation. In this case, incubators, pumps, valves, controllers, all integrated into a platform that is about 48 cubic feet, about the size of your average kitchen refrigerator. But in talking with SpaceX, who is the payload, of course, deliverer into the station, their requirement to bring a payload up into space is the size of a shoebox, or 1.6 cubic feet. And so the challenge was to engineer the platform such as the size of a kitchen refrigerator to the size of a shoebox. And so fortunately, we were able to work with space implementation partners that are expert payload developers and engineers, such as TechShot, Space Tango, and Bioserve, and actually was able to reduce this instrumentation to a size that can be flown by SpaceX. And so we had, in fact, done that several times over the course of 6 years, starting in 2018. We have a series of launches with SpaceX, beginning SpaceX 16, all the way to the more recent one, SpaceX 27, which launched March 14 of this year. And shown here are astronauts who've been working on tissue chips and the various platforms up in the space station. And they normally stay up in space under microgravity for a period of 30 days to 45 days. So the question then is, what do the results look like? So if we just look at one of the projects looking at immunosenescence, so in this case, looking at the effect of microgravity on immune cell aging, we can show that in just 15 days, instead of years here on Earth, we can already see changes in some of the cellular markers for aging. So in this case, an example is prostaglandin D2 receptor, which is a marker for senescence and proliferation and wound healing. So G stands for ground and F for flight, showing that prostaglandin D2 receptor do in fact show a huge increase in expression within 15 days of flight, showing again that we can capture some of the cellular changes as a consequence of things that mimic aging. Looking at another project on cardiac dysfunction, you can see here too that microgravity within 4 days already induces a very specific decrease in twitch force as well as contractility. Again, showing here that we can capture some of the aging effects. So in fact, the heart cardiac dysfunction, 6 months of exposure in microgravity is equivalent to 10 years of biological aging here on Earth. So we can essentially and effectively be able to capture some of these changes in a very short period of time. Switching gears now and looking in terms of precision medicine, we have launched a new program starting in 2021 called Clinical Trials on a Chip to inform clinical trial design and precision medicine. So the goal here is to incorporate patient diversity, the various mutations, various ethnicities, sex into the chips to be able to capture the heterogeneous patient population and in some ways establish guidelines or criteria by which we can establish clinical trials and inform clinical trial design by establishing patient recruitment criteria, being able to stratify the patient group, which are most likely to be able to respond to the treatment, and those and identify actually those who are also most likely to develop adverse event, and also to develop surrogate clinically relevant biomarkers. It's a biphasic program, so the first 2 years was to essentially capture the pathophysiology of the disease by taking patient-derived induced pluripotent stem cells, putting them on chips, and being able to interrogate them for markers that would show proper mimicking of the pathophysiology of the disease. And then phase 2, which is the current phase of the program right now, is to be able to treat the chips with candidate drugs that are currently being tried in human clinical trials, and in parallel to show that we can capture some of the, or if not all of the changes being seen in the conduct of human clinical trials. So in that regard, we have a number of funded projects looking at non-alcoholic fatty liver disease, atopic dermatitis, muscular dystrophy, progeria, which is a premature aging rare disease, tendon inflammation, ALS, heart diseases, PKD or polycystic kidney disease, and then also looking at preterm birth and things that causes preterm birth by mimicking fetal-maternal interface, and then prostate cancer metastasis to the bone. Switching gears again, so in terms of how do we build confidence into this technology, again, this was technology that's been about 10 years of age in terms of its development, but how do you build confidence in the technology such that the community can start working on it and using it. So it's essentially a three-stage validation framework that NCATS stood up. Started a program. We call it the physiological validation, which is, can the tissue chip developers mimic organ function and structure, and test them on a training set of reference compounds similar to the hepatotoxic compounds that I mentioned about in the example to the liver. So we have reference compounds for each of the different organ systems, and certainly this has resulted in a number of publications for the community to vet and look at and be able to reproduce. But also, we also wanted to, at the NCATS, wanted to create an analytical framework where we stood up independent testing centers who have not worked on chips before, and asked them, can you transfer the technology to these testing centers, and can these testing centers essentially reproduce and demonstrate robustness and reliability of the data that was published by the tissue chip developing centers. So in that case, we funded MIT and Texas A&M as the two testing centers, and then also established a database at the University of Pittsburgh. And then in the last stage of the validation framework is called the industrial. And this is where we have industry and regulatory agency look at the data that's being deposited in the MPS database, and to show the robustness of the results, and thereby leading them to use and test these platforms with their own proprietary set of compounds, or to ask some of the CROs to start working on the system. So in this case, we encourage MIT and Texas A&M to develop their own self-sustaining business model. So MIT became Javelin Biotech, and Texas A&M became a testing consortium, and the University of Pittsburgh MPS database is now called the Biostatistics Analytics Platform. So in terms of the database, again, this is a federated database that is free for everyone to look at the data. So it takes in tissue chip data and incorporates that and integrates that with clinical data sets as well as in preclinical data sets, and being able to look at a number of different compounds that's all been tested in humans, and being able to develop analytical tools that would show modular reproducibility, compound safety and testing, as well as mechanism of disease and progression. At the NIH, we have also encouraged the spin-out of technologies into the commercial realm. So there's a number of startup and spin-off companies, all operating around tissue and organs on chips, as well as organoid technology. And this is about 30 or so of them. And so we have supported some of these companies, and they are providing platforms and consumables as well as CRO-like services. Then the question would be, in terms of regulatory acceptance, what is the FDA looking? How is the FDA looking into this new technology? So the FDA actually has formed an FDA Alternative Methods Working Group that is able to look at this new technology and be able to evaluate it within its laboratories. And they also stood up a program called the iSTAN program, which is able to take MPS or tissue chip data as a standalone data in order to be able to be used for IND submissions, investigational new drug submissions. More importantly, in December of last year, a bill was passed called the FDA Modernization Act 2.0, which essentially contains language that struck down preclinical tests that includes animals and inserted non-clinical testing. So in some ways, what this does is it opens up the submission of data sets from in vitro, including microphysiological systems, as well as in silico and in chemical for submissions to the FDA. And so what this does is enable drug registration without the absolute requirement for the use of animals in safety toxicology assessments, especially where alternative risk assessment tools are available. And there's also signals that there is increasing recognition that the animal-based safety and toxicology and metabolism studies have limited predictive value, and shows that growing evidence that new alternative methods such as tissue chips and microphysiological systems can provide equal or more valid data sets in terms of predicting human response. And so in this particular regard, we have worked with the FDA to qualify some of these tissue chip platforms to qualify them as drug development tools. And here's the tool qualification process for the FDA. There's a letter of intent stage, the submission of a qualification plan, then the full qualification package, and the qualification recommendation. And all of this has to be within a specific context of use. And so in this regard, we had worked then with the FDA and signed a letter of agreement between NCATS and FDA with the goal of having microphysiological systems be qualified as a drug development tool for a specific context of use. And so with this, we are going to set up between 3 to 4 centers. We call them Translational Centers for Microphysiological Systems, or TRACE MPS. And each center that will be funded is tasked to qualify at least 4 to 5 microphysiological systems, each with a different context of use. And we should be able to announce which of the centers will be funded by the end of this year. In terms of pharma uptake, we are looking now, working with, as I mentioned, with the IQ MPS affiliate, about 24 different pharma companies. They have already published a number of publications in terms of fit for purpose for what industry would like to see these tissue chips be developed for, so that it can be used for industrial purposes. And so they have guidelines in terms of liver, kidney, lung, GI, skin, and cardiovascular conditions, with more coming up in terms of additional publications. Where they have been being used, shown here are the pharmaceutical companies working on tissue chips and the particular commercial platforms that they're using, the areas of application in drug development, and the type of tissue chips shown here on the first column, from blood vessels, bone marrow, gut epithelium, liver, and primarily looking at the use of tissue chips for target identification, lead optimization, preclinical safety, preclinical efficacy, and pharmacokinetics and toxicokinetics. So from my presentation, I think you will get a sense that tissue chips can essentially be deployed and used in all areas of drug development, starting from target identification and validation, through lead optimization and identification, preclinical safety, and clinical assessment. And the last thing that we're trying to do is to also engage next generation scientists, engineers. And so what NCATS did last year is to work to establish the creation of an international forum that investigators working in tissue chips can essentially connect with each other, exchange data and resources, as well as to educate the next generation scientists, engineers. And so we had a meeting last year in New Orleans, this year in Berlin. And just to show you on the chart that there's a growing interest and presence of in this new technology. So in New Orleans, we had 450 attendees, in-person attendees. In Berlin, there's almost 1,300, almost certainly doubled the number of abstracts from 318 to 756. Invited speakers were 142 and 166. We had a big increase in number of posters from 189 to 553. Importantly, looking at early stage investigators or young scientists growing from 187 to 459, as well as the number of countries represented, 26 and 39. So the next meeting will be in Seattle next year, June 26 to 30. And lastly, we also form an international microphysiological system society with a goal to accelerate the use of MPS to improve health, welfare, and the environment, and to train the next generation of scientists. And with that, I'll end with a summary showing that we have been working with the developers and suppliers in terms of developing the technology, working with the end users such as pharmaceutical and industry in terms of identifying context of use and fit for purpose, and certainly working with the regulatory agencies in terms of developing standards, validating systems, as well as qualification. And then we'll end with just an acknowledgement in terms of the partners that we have, starting with within the NIH, there's about almost 60 or so individuals, too many to name individually, but just collectively, a number of them, especially people that I work with closely, Pasley, Dimitri, and Chris, and certainly our partners at with the FDA, the International Space Station, the Center for Advancement Science and Space, NASA, the IQ Consortium, and I did not mention our collaborations with BARDA and VA, but they are also partners in this regard. And so with that, I'll end my presentation and happy to take any questions if there are any.
All right. Thank you very much, Danilo, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just submit it into the Ask a Question box located towards the bottom of your screen. All right, let's go ahead and get started. Okay, the first question we have here is, for the COVID infectivity study, 7 drugs looked promising in 2D, 3 looked promising in 3D. What do we understand about the predictive nature of this 3D data on COVID therapeutics from NHP or human studies?
Yeah, so that's an interesting question. Yeah, so certainly, with the predictiveness, it's been shown that at least in the human trials that is ongoing right now, that it seems to be quite more effective. Certainly if we follow the lead with the 2D cell culture system, would be probably chasing some of those other antiviral drugs that may not show promise after all in human trials, because the 2D cell culture system are just monolayer Has7 cells, whereas the 3D has the biomechanical breathing in and out of the lung epithelial cells, so the stretching the lung epithelial cells, which turns out to be an important factor in being able to mimic the physiology of the lung in the sense that they're able to secrete surfactants and other things when it undergoes stretching. And so it's much more physiologically relevant.
Perfect. Thank you. All right, we have another question here. What are the strengths and limitations of MPS over whole body animal in vivo models? And what's the best way in selecting the best model for the research question?
Yeah, I think the advantage certainly of microphysiological systems over in vivo is the use of human relevant cells and tissues. Obviously, one can also make humanized animal models, but that takes a lot of time and resources to develop those. And so in some ways, looking at individual organs in a humanized animal model is somewhat the same as using human cells and tissues on microphysiological systems. I mean, obviously, we're still far off from recreating an entire body system on a chip. We don't have all the other organ systems, but certainly I think with the 10 major organ systems right now, we have some of the relevant ones that mostly are looked at in terms of drug development.
All right, thank you very much. On to the next question here. Where are the current methods inefficient and or flawed, and where can 3D models fill in?
Yeah, the current models, as I've shown, are not necessarily flawed, but probably deficient in being able to properly represent the human response. I think people in the preclinical area would say there are probably more mice cured of cancer than actual people, because we can show effectiveness of certain candidate drugs in animal models. But the true nature of it is, humans are not animals, and animals certainly are not humans. And so while they are effective model system for certain conditions, they do not necessarily all recapitulate the human response. And so it becomes a little bit more difficult to take the findings from some of those animal models and invest a lot of time, money, and resources to bring them to trials, only to find out that they may fail. So I think what we're trying to show here is that we can use microphysiological systems and tissue chips to be able to properly represent human relevant data, as well as be able to generate data that would hopefully inform and be more predictive of human response. And in a number of different studies, animal models too are a very inbred group, and the reality too is we're a very heterogeneous human species. And so part of the effort that we're trying to do with the chips is to be able to represent each and every individual on the chips. And so there will probably be a time in the future when in the course of precision medicine, where each and every individual can have their own chip. We call it the U on a chip. That can be used to inform, prognostically, which, how the disease is going to happen, what sort of treatment a particular individual has, would likely benefit from if there's a number of candidate therapies. So it eliminates some of the issues in terms of identifying what is the best and most efficacious treatment for a particular individual. We're talking about at the individual level.
All right, great. Okay, next we have another question from an audience member here. How much efficiency can we expect from studies on tissue chips rather than using any other animal models?
So in terms of efficiency, I think, how do you measure efficiency? It's more of what percent of, I'd say it's what percent of drugs actually make it into the clinics and successfully be going and being used successfully without that being withdrawn. So we're still at the early stages right now I think this year is probably going to be a big year for tissue chips, in the sense that a number of candidate drugs are now being submitted for IND. So probably too early to tell right now in terms of efficiency, but certainly from some of the data that we can see, just looking retrospectively in terms of what has been predicted by tissue chips in terms of human response versus the failure of animal models that have not been able to predict the human response. I mean, I gave the example of the liver on a chip in terms of predicting the hepatotoxicity. So that shows, certainly a remarkable sensitivity of 87% compared to 47% in 2D cell culture system.
Perfect. All right. It looks like we have one final question here. So we'll go ahead and wrap up with this one. What do we anticipate as the future on 3D tissue models?
Well, I think the future certainly is very promising. There's a number of efforts internationally, not just in the United States, but certainly Europe, Asia, and Australia, and South America, and being able to advance tissue chips as what's called new approaches methods, which hopefully will complement, if not replace, animal models. So there is a big concerted effort internationally, and so I think that will just lead to more support, as well as wider and more like global acceptance and use of this technology in the course of doing safety studies, in the course of doing efficacy studies, as well as being able to deploy them more and more and closer to the clinics. We're talking about pairing up tissue chips with individual digital twin technology to be able to not only use computational models to predict human response, but also to validate those computational predictions with tissue chip technology as well. So I would say the future is very promising.
All right. Thank you very much. Thank you again, Danilo Tagle, for your time today and for your important research. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until September 20th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you, everyone.